You're at the cutting edge of RNA therapeutics. I'm John Cooke, Chair of the Department of Cardiovascular Sciences and Director of our Center for RNA Therapeutics. And today we're going to be talking about lipid nanoparticles and delivery of RNA. Joining me in the studio to introduce our guest speaker is Dr. Kristopher Brannan, one of our faculty in the Center for RNA Therapeutics. Dr. Brannan?
Thanks. Dr. Vartant Shah will also be joining us online as a discussant. Dr. Vartant Shah is an instructor here at Houston Methodist working on RNA therapeutics as well. And so, it's my pleasure to introduce Dr. Dominik Witzigmann from Nanovations Therapeutics. And so, by way of background, Dr. Witzigmann got his PhD in pharmaceutical technology at the University of Basel in Switzerland. Since then, he's worked on projects at the University of Basel and targeted nanomedicines and DNA delivery technologies. At the University College London on toxicity, RNAi and oncology at the German Cancer Research Center, and mRNA-based genome editing technologies at the University of Zurich. He's recently joined Peter Collis's group at the University of British Columbia to focus on RNA delivery using lipid nanoparticle or LNP technologies. And he's developed quite a track record in developing nanomedicine innovations for nucleic acid deliveries. He's served as a leader in the Nanomedicines Innovation Network, where he founded and led the NanoCore, which supports dozens of research projects focused on advanced nucleic acid delivery technologies. He has recently, to translate these discoveries into the clinic, founded and leads the Nanovations the LNP nucleic acid company Nanovation Therapeutics, where he's innovating LNP technologies enabling RNA delivery beyond the liver, which he's going to be telling us about today. And before you sort of launch into your presentation, I was hoping we could have a brief discussion about your path, sort of how your path led to mRNA therapeutics, which are near and dear to our heart here at the Center for RNA Therapeutics at Houston Methodist. And where you think, I guess, LNP mRNA technologies are headed maybe in the next 5 years, both inside and outside of the vaccine space.
Yeah, thanks a lot for inviting me, and happy to provide a bit of background. And I'm a pharmacist by training, did my PhD in pharmaceutical technology. As you mentioned, then was at the Cancer Research Center, where I had focused on microRNAs and long non-coding RNAs, where I learned a lot about the RNA biology. And then by moving and joining Peter Collis, working on genome editing, really combining the basics of RNA inside a cell and using that for therapeutic applications. And it was really driven by my background of being in pharmaceutical technology and applying the learnings from RNA biology to therapeutics. And to your point of where I see the next 5 years, I think it's a super exciting time because with the advent of mRNA and gene editing, it is coming true that you need a delivery system like LNPs or other non-viral delivery systems to enable such therapies. And beyond vaccines or beyond infectious disease vaccines, I'm super excited about all the developments on cancer vaccines and personalized therapies, as well as genome editing. So within the next 5 years, I strongly believe we will see more personalized cancer vaccines, as well as genome editing applications, either for the liver, but even beyond the liver, and even thinking about local administration. This is what I see in the next 5 years. Beyond that, obviously, it will go into the direction, I strongly believe, of personalized medicines, using all the omics diagnostics and applying that to therapies which can tackle the genome.
Yeah. Dr. Witzigmann, so nice to have you on the show. And as Dr. Brannan mentioned, we're very interested to hear your latest take on lipid nanoparticles and how we're going to be delivering mRNA in the future. But before we launch into your talk, let me just tell our viewers at home that they can send in their questions to you. Two ways to do that. If you're at your computer, go to pollev.com. That's POLLEV.com. Enter DeBakey, and then you can answer, you can give us your questions. Alternatively, you can join us by text. You can phone in at 37607 and text DeBakey. That's 37607, text DeBakey. And then just let us know your question, and we'll ask Dr. Witzigmann. So with that little done, housekeeping done, please, Dr. Dominik Witzigmann, tell us what your take is on lipid nanoparticles.
Thanks a lot. And what I will tell you about today is about using lipid nanoparticle technology to enable RNA therapeutics beyond the liver. I give a brief outline of like where we have started, and then what we at Nanovation Therapeutics focus on, and like what is one of the hot areas in the gene delivery field in general is like how to get beyond the liver and how to overcome this nucleic acid delivery barrier. I first would like to start a bit like where we are currently at. We have seen the the COVID-19 vaccines, the liver applications which are currently in the clinic or which have been approved for delivering siRNA. But when we look at our body, there are so many different organs, and we currently have more than 4,000 preclinical developments and more than actually like 2,000 clinical programs running in the field of RNA therapeutics, which includes mRNA as well as siRNA gene editing constructs. So it's a super exciting field, and we will see a lot of interesting developments over the coming years with the focus on various different areas. And like each delivery technology as well as like nucleic acid payload technology will find its niche application where you either silence a gene, express a gene, or edit a gene.
Dr. Witzigmann, do you want to share your slides with us?
It should be sharing, actually. It says it shares. Give me a second. Sorry for that. And I go back. Let me know if you see it now. Is it in full mode?
It's coming. Here we go. Perfect.
Perfect. Sorry for that. We shared already in the preface of setting this up. Yeah, this is what I was talking about, like all the clinical development ongoing and all the different organs where we currently see RNA therapeutics, either as naked, as RNA, or also like mRNA applications, as well as like, let's say, siRNA conjugates, as well as like non-viral delivery systems like LNPs.
Dr. Witzigmann, you don't have numbers associated with that graph you just showed us, but I think it's probably in the range of about 200 programs that are in development right now. Is that about right?
It's about right. There actually, like when we look at the entire like RNA field and like including siRNA conjugates as well as like mRNA gene editing, we are actually close to 2,000 clinical programs actually.
Wow.
So we have access to like an intelligent database called Beacon, and there you could see that there are actually close to 2,000 clinical programs in the pipeline at the moment. Many of them cover the same aspects, but different angles of the disease. So when we go back to the topic of today, the lipid nanoparticles, obviously everyone should be aware of the breakthrough during COVID when the lipid nanoparticles have been used for mRNA delivery intramuscularly to induce an immune response. But obviously this technology has been there before and like culminated in the approval of Onpattro in 2018, which was the first ever siRNA therapeutic, which delivered an siRNA to the liver for treating a genetic disease called transthyretin-induced amyloidosis, where you knock down TTR in the liver and therefore get an efficient treatment of the patient. When we compare what is the composition of Onpattro, what is in the COVID-19 vaccines, just to, let's say, focus on these three applications or these three lipid nanoparticles, all of these have an ionizable cationic lipid, all of these have a phospholipid like DSPC, all have cholesterol, all have a PEG lipid to shield and especially stabilize during storage, and all of them have an RNA payload in different ratios of RNA to lipid ratio. And what is astonishing here, so the difference between these different formulations was the ionizable lipid, or is the ionizable lipid, as well as then the route of administration. So when we now look at how these particles look like, which will be important then when we look at like technologies for delivering beyond the liver, when we look at siRNA LNPs like Onpattro during the formulation process, when they are dialyzed, then after the let's say, either microfluidics or using T-junction mixes, and you dialyze and purify the LNPs into physiological buffer, you end up with these solid core particles. With the mRNA, they often have like these blebs. But what is becoming clear that when you inject these intravenously, these 4-component conventional systems, they are really effective in treating liver diseases. And this is what we have seen with the treatment of Onpattro, for knocking down TTR in the liver and therefore enabling a treatment which hasn't been there before, for treating patients with transthyretin-induced amyloidosis. But now the question is, it's really effective for the liver and currently is in studies for genome editing or base editing, or also applied for BRAM editing in the liver. When we look at the circulational half-life of Onpattro in mice and think about like how long does the LNP actually circulate, and is it like 15 minutes, is it like several hours, or even a like day? It actually comes down to like it's 15 to 30 minutes. So there's a really quick elimination from the blood following an intravenous administration, and then a high and fast accumulation in the liver. And therefore, like we see a strong expression or silencing of the liver, but obviously that will also make it difficult to target tissues beyond the liver because most of the dose ends up in the liver. So what have been traditional approaches to that? As outlined at the start, most of the current LNPs approved, they have a PEG lipid with a C14 lipid chain, which anchors the PEG, the polyethylene glycol, in the LNP, but following intravenous administration, there's like a protein corona formed with different proteins like ApoE, and therefore following the intravenous administration and desorption or adsorption of ApoE to the surface of the LNP, it will bind to the LDL receptor in the liver and therefore efficiently get into hepatocytes in the liver. So approaches to overcome that have been the inclusion of PEG lipids, which have a longer lipid anchor and therefore don't dissociate as quickly as the C14 chain. And therefore you can significantly increase the half-life of the LNP in the blood and therefore decrease liver accumulation. The problem with that is that the more PEG you put on the surface of an LNP, the lower is the transfection potency. So you run into the so-called PEG dilemma that, yeah, you want to increase the circulation half-life, but you actually hinder the transfection of cells. And which is outlined on this left graph, that with the increasing amount of PEG, you limit actually the transfection, which is outlined here for as RNA, so you don't have any knockdown if there is too much PEG on the surface. The second part is if you think about the intravenous administration, there's a lot of discussion ongoing in the field about immune responses against the surface of the LNP, and therefore issues with repeated administration when it comes to these stable PEG conjugates on the LNP. So the main topic of today is like how can we actually overcome this delivery challenge and like deliver nucleic acids to extrahepatic tissues? And one clear statement from my time working with Peter at the university as well as starting Nanovation Therapeutics and what we have seen from COVID versus now, there is no one fits all. So the LNPs have worked really well in 2018 for Onpattro and liver application and siRNA. They have worked really well for COVID-19 and mRNA for vaccine applications, but it's not easily translatable to different organs, different payloads, and also a different route of administration. So what has to be considered is like, what circulation half-life do you need? And if you want to target tissues beyond the liver, you need longer circulating systems. They need to be transfection potent, so you should not shield the LNP that they lose their transfection potency, as well as you need to think about not only efficacy, but actually safety. And so the therapeutic index here is like key to all the different applications. As mentioned, like I'm the co-founder and CEO of Nanovation Therapeutics. We are headquartered in Vancouver. One of our founding members is like Peter Cullis, who is often seen as the founding father of LNP technologies. And our focus is really the LNP development. And some of the data I will show today is focused on how we overcame this nucleic acid barrier and came up with new LNP types to have longer circulation half-lives and therefore target extrahepatic tissues. A key to that is looking at the different components. So when we look at the COVID-19 mRNA vaccine development, the traditional way of like developing these drugs was to take components and composition and modifications from different suppliers or different biotech and pharma companies, versus like when you look at the different components, you have the specialty ionizable lipids as the most important component to entrap and encapsulate the nucleic acid and improve the efficacy as well as safety profile. Then you have the LNP composition, which is key for any application, especially when you think about route of administration, You have surface modifications, so you can play around with different modifications in terms of the PEG, or as well as targeting ligands to improve the specificity, or also then about the RNA payload. An LNP can only bring a certain specificity towards an application, versus the RNA can also embed specificity to a certain cell type or a certain gene sequence. When we apply the lipid nanoparticles to extrahepatic tissues, there are three different options. One is apply the LNP to ex vivo applications. Currently, obviously, like electroporation or especially viruses are used in the T-cell model engineering ex vivo, but LNPs are actually really potent, and you could modify T-cells ex vivo and then inject them back into the patient. Second application would be locally. We have seen with COVID-19 the local administration into the muscle, but you could use this local administration of LNPs for muscle applications, but also think about tissues or organs like the eye, or also like tumors, where you can use this administration route. What I will focus on today is the systemic administration. So how do we get to certain cell types, like the T cells, following an intravenous administration? I want you to think for a second, like which extrahepatic targets, like organs or cells, you find most interesting. I will only cover a portion of the applications, obviously, today, but happy to then talk about and discuss where are the hot areas, where are maybe areas where it's really difficult to overcome the delivery barrier at the moment. So please keep that question in mind. So now stepping into what we call the LC-LNP, the long-circulating LNP technology. And the name already tells you it has something to do with the blood circulation time, what we have developed. And this stems from the decades of research in the liposomal field. So when we look at the small molecule delivery of for example, anti-cancer drugs, and using liposomal systems like Doxil, this is a pegylated liposome for delivering doxorubicin, these type of lipid-based nanomedicines have really long circulation half-lives, which then were used for cancer therapy. And here's this landmark paper from Harrington et al., where they could show that by this long circulation half-life, you actually get accumulation of the liposomal system in Kaposi sarcoma, and therefore enhanced accumulation of the small molecule drug. And then we have seen other small molecule drugs which use different helper lipids like swingomyelin instead of DSPC. And then we came to the Onpattro development, where we looked at the siRNA delivery to the liver. So the question was, what can we learn from the liposomal field? And when we look at, for example, in this example, an siRNA containing LNP, I told you at the start they have these solid core structures. We looked at the liposomal development and understood that these conventional LNPs have mostly a monolayer of lipid, whereas when you look back into the developments in the 90s, where the liposomal systems really came into the clinic, they have lipid bilayers. So can we actually apply this learning and change the morphology of an LNP towards more like a liposomal system, which then has a lipid bilayer, which should impact the circulation half-life, because there should be less accumulation of proteins or certain proteins, and therefore also enable longer circulation in blood. And what you see, you don't have, these are cryo-EM images, you don't have to be an expert to see the huge difference between a conventional LNP, which is the solid core structure, versus what we call these fried egg structures, where you have a clear lipid bilayer and a condensed core. And this really comes
show us, Dr. Witzigmann.
Yeah.
These are empty LNPs. These don't contain mRNA, I guess.
In this image, these are siRNA containing, but the mRNA containing actually look pretty similar.
Okay, so with mRNA, they would look similar.
Correct. Yeah.
Okay.
So what we did here is looking at the different, you remember that one of the components is, for example, phosphatidylcholine, or one of the helper lipids. So if you increase the helper lipid content in a lipid nanoparticle, and typically you have in the Onpattro formulation, or also the Moderna or BioNTech Pfizer vaccine, you have roughly 10% of DSPC and 38.5 or 40% cholesterol. If you increase the helper lipid concentration, you see that you get from this solid core structure towards more this fried egg, or then even double bilayers, which will then also impact the the entrapment, and you see when you overcome a certain like percentage of helper lipid, in this case with a specific ionizable lipid and a certain lipid to RNA ratio, you lose the entrapment of the RNA. So one of the first steps was to analyze the pharmacokinetics of the systems which we had developed. And when you inject a conventional 4-component system with siRNA or mRNA in the typical ratios, you have a really quick liver accumulation. What you see here is a SPECT-CT image where you see like a clear accumulation of the LNP in the liver, versus if you inject these so-called LCLNP, these long-circulating LNPs, you get a completely different distribution. And what you see is the anatomy of the heart, liver, spleen, like highly perfused organs, which already indicate an increased blood circulation half-life. So when we analyzed then the percentage of injected dose of the LNP, but as well we looked at the payload like mRNA or also like DNA, you can see that you can try the circulation half-life from half an hour towards several hours, like 4 to 8 hours in this example, depending on the molar ratios, but also depending on the helper lipid. Obviously, when you do this detargeting of the liver, you overcome one aspect, which is the liver toxicity. So when you inject a standard conventional system which accumulates quickly in the liver, you also get, even so it's like transient, you get high liver biomarkers, indicated here by the ALT, AST levels, which are diminished, and we even went up to like 10 mg per kg without having elevated liver biomarkers. And you also don't see this when you do a repeated administration. And here we did like 4 injections at 1 mg per kg. So now this was kind of like stating that, yeah, we have an enhanced circulation lifetime. So how can we now look at the efficacy? What has been done in the field is like using the standard conventional LNP and using different ionizable lipids, and therefore looking at the efficacy, and in this example having an mRNA encoding luciferase, injecting the LNP intravenously, looking at the relative luciferase activity in the liver, and like 2 of the benchmark lipids like ONPETRO, or the MC3 in ONPETRO, or ALC 315 in the BioNTech Pfizer formulation, Comirnaty. There's a significant difference in terms of efficacy when you compare ALC and MC3. We have developed our own lipid library and screened these in lipid nanoparticles targeted towards the liver, and have done this in mice as well as NHP. But we also then looked at like how can we use these ionizable lipid in the LCLNP. And there's one interesting factor that, please focus first on the left-hand side. If you take the same ionizable lipids, in this case MC3, and you look at a conventional 4-component LNP or an LCLNP, and you look at the expression in the bone marrow, first of all, by changing the pharmacokinetics, you can like increase the accumulation in the target tissue, and therefore also significantly increase the expression in the bone marrow. And when you now switch out and combine it with different ionizable lipids, you see here an interesting effect that if you compare now MC3 or ALC and an LCLNP, there's not too much of a difference. So you can further enhance the efficacy by fine-tuning the ionizable lipids and therefore enhancing the efficacy in the bone marrow. And this is the main principle we use to target extrahepatic tissues, that we always start off with an LCLNP. We have different ionizable lipids from our library, and then looking at different applications. And what I've outlined here, just like 4 examples for different tissues and cell types, where we have looked at different programs internally as well as with partners. For example, where we looked at how can this LCLNP be used to target HSCs versus T cells. And we see that there's, independent of the, let's say, immune cells shown here, the stem cells, we see significant improvement in transfection potency when we use these long circulating systems compared to a conventional 4-component system. Same applies when we look to the bottom part, like when you look at monocytes as well as macrophages in spleen or other tissues, we see a significant increase in transfection potency, as well as dendritic cells in lymph nodes. To summarize this part, and before going to the last section, then also a discussion part, is what we know, the conventional systems, they typically have a 4-component system, a typical ratio of like 50% of ionizable lipid. They are excellent in targeting the liver. They have a rapid liver accumulation. Depending on the let's say application we have seen in the clinic with some of the news like base editing data, that is obviously a limited therapeutic window. And also the IP space is one factor which plays a huge role in this space. There are other approaches. There are, for example, like the targeted approaches where you add a fifth component or you add targeting ligands. One thing to keep in mind, the ionizable lipids have a significantly decreased or increased safety profile, a decreased toxicity compared to permanently charged cationic lipids. Then if you modify these standard LNPs with charged lipids or also with targeting ligands, you have to keep in mind that there's a quick elimination, potentially a quicker elimination via different organs, and the manufacturing complexity, especially when you talk about targeting ligands, have to be kept in mind. So where we apply the long-circulating nanoparticle is really about using these for intravenous administration for tissues beyond the liver, increasing the efficacy as well as the safety profile because of the detargeting of the liver, and looking closely, this from a company perspective is obviously really important, look at the IP of the LNP composition as well as the ionizable lipid. One question which always comes up, what about like modifying an LNP with targeting ligands like antibodies or single chain or peptides? We have done internal studies as well as with partners where we modified the LCLNP as well as conventional LNPs. You can increase the selectivity, and let's say our conclusion from the studies we have done is that the LCLNP is a better base formulation because you first need to have a long circulating system that the targeting ligand first can actually like bind to the target cell. Still, you always have to keep in mind this manufacturing complexity. So giving a short outlook, what we had already in the discussion at the start is, what excites me at the moment is that nucleic acid therapeutics have a lot of potential for different organs, different applications, and especially with the longer RNAs like the mRNAs, circular RNAs, self-amplifying RNAs, you need a delivery system. Like for siRNA, we have seen a lot of development on the conjugate side, but for mRNA, this is not possible. And therefore, using delivery systems will be the key to overcome many of the critical hurdles. During COVID, there have been like estimations, where does the market go? And obviously now the COVID-19 mRNA vaccination rates go down. But there was an interesting statement by Ugur Shahin from BioNTech during COVID, who said that, 1/3 of all therapeutics will be nucleic acid based in 15 years. I still strongly believe that nucleic acid therapeutics will play a critical role going further into the future, where we use the technology to not only prevent diseases by using it for vaccine applications, but also actually be able to cure diseases. So to summarize, when we look at the different applications, how to target extrahepatic tissues, and we look at the ex vivo application, local, only covered systemic administration, one important aspect I mentioned that the one-fits-all approach does not work, and the approach we take, but many in the field discover at the moment, is this, what I like to say, fit-for-purpose development. So you look at the target product profile and say, what is the target tissue? Okay, it's the bone marrow. What is the route of administration? Okay, it's intrafemoral, or is it intravenous? You look at the payload. Is it an mRNA? And you want to have longer expression, or is it like a genome editing application where you want to use a hit and run approach? You want to go in, do editing quickly, and go out to limit off-target effects. And then also, like, as said, like the application. Is it genome editing, gene silencing, gene expression? The tissues we are currently working on internally as well as with partners are focused mainly on stem cells and immune cells, and here I outline some examples where we look at blood, spleen, bone marrow. One of the areas we are really interested with partners is to look at tumor therapy, how to apply the long circulating system to target actually, like, for example, tumor resident macrophages or also cancer cells. And working on the nucleic acid payload and then applying or changing the LNP composition to a certain ratio depending, for example, on if it's an siRNA versus an mRNA. And also when you think about DNA, there are other hurdles to overcome, like the nuclear pore membrane. So with that, the last two slides is really the ask to you, or like, do you believe that delivery technologies are key to enabling the genetic medicine revolution? I stated that I strongly believe that delivery systems are key to that field, and they will be key. And I hope that when you shortly thought about this question that, yeah, your answer was also yes, and that you see the impact delivery technologies can have in this field. And with this, I thank you a lot, and yeah, looking very much forward to a fruitful discussion.
Thank you, Dr. Witzigmann, for that nice presentation, and you certainly have been one of the pioneers in this area of nucleic acid delivery. And now we have some time for discussion. Before we begin that discussion period, let me just remind our visitors online that you can write your questions into us. Just text 37607 and text DeBakey to that number, 37607. Alternatively, if you're online, if you're your computer, go to pollev.com, that's POLLEV.com, and enter DeBakey. And I think what I'm going to do now is turn to my colleague here and ask Dr. Brannan to moderate the discussion.
Sure. I don't think we have any questions from viewers online yet that have been posted for me to see anyway. So, yeah, I can open with a couple of questions. I guess for personalized approaches, how much as Nanovations are you thinking about sort of modifying the nucleic acid payloads to be more responsive to the context of the cell or the tissue that you're targeting, as opposed to the LNP delivery vehicle itself?
It's a really good question, and I think it's key to not only look at the LNP but also the payload. So what we have done internally, especially when it comes to applications of the bone marrow or like immune cells in extrahepatic tissues, is to look at RNA modifications, like chemical modifications, as well as like the UTRs and caps, to make it more specific. And what we have seen is that certain modifications of the mRNA actually enhance the extrahepatic to liver ratio. So we can further boost the safety profile of these LNPs, despite influencing the pharmacokinetics biodistribution of the LNP, but by purely expressing higher levels in certain tissues. There are several companies in the field working on like further modifying RNAs and making them more specific, and I think it will be key to actually break some of the applications and enable certain cell applications, because safety is a key aspect which has to be considered.
Just to follow onto that, I just want to congratulate you for developing LNPs that aren't so trapped by the liver, because that's been the major problem, is LNP targeting the liver and get very little getting elsewhere. So the long-circulating lipid nanoparticle looks very promising. And then you mentioned some other things that you could potentially do to increase preferential tissue distribution, changing the lipid composition, maybe adding targeting antibodies to the lipid nanoparticle, or as Dr. Brannan mentioned, you can also modify the mRNA so it's preferentially expressed in the tissues. So it's really kind of a multi-level way to get to where your target is, isn't it?
Yeah.
One point I wanted to ask you to address there is, when you're talking about tissue target, when you're giving the LNP systemically, aren't you really talking about endothelial targeting? Because for the LNP to get into a tissue, it has to bind selectively to the vessels that subserve that tissue, and that's the endothelium. That's going to be what you first encounter. So you must be targeting differences in endothelial expression for that preferential tissue targeting.
To some extent, yes. And maybe I start with explaining like how we typically screen the different LNP technologies. We have like typically two steps when we use reporter genes. One is at the tissue level, where we use mostly reporter genes like luciferase, where we just look at bulk tissues like spleen, bone marrow, heart, lung, kidney, etc. And then at the second level, we look at reporter genes where we can use flow, for example, to distinguish different cell types. So we see, for example, when we look to the bone marrow, that we can efficiently hit stem cells and like long-term HSCs, for example. Key is there that when we look at, for example, the bone marrow or the blood compartment, as well as tumors or the spleen, these organs have all fenestrated tissue. So the LNPs are able to get out of the blood vessel and therefore accumulate and target like the cell types of interest. Versus when we look at tissues like the kidney, there I would give you right that with the kidney or other organs which have really tight blood vessels, there's much more difficult to get out of the blood vessels into the tissue and then target other cell types beyond the endothelial cells.
We have a junior colleague on the line, Dr. Vruthan Shah, who has a question.
Yes. So I wanted to follow up on since we are RNA therapeutics, so what do you think in the next few years, how do you see with the emerging technology of RNA, how can your system be incorporated into that? And the second question I have is that what are the hurdles do you see, even though you made it's going not trapped by the liver, but going to the other organs, which other organs do you see is hard to target with your technology?
Mm-hmm. So starting with the first part, where I strongly believe, and obviously biased perspective by the technologies we developed, is in immune cells, and I would say that's not only a focus from us, but also like other LNP developers, as well as like pharma and biotech, thinking about like in vivo T cell engineering. How can we do an intravenous administration, target T cells and engineer the T cells, or like also at macrophages. Tumor is definitely getting hotter when it comes to RNA therapeutics. Or then stem cells. So there have been like a lot of publications about like targeting stem cells with antibody modified, for example, or single chain modified lipid nanoparticles. We take a different approach. We always try to keep the system as simple as possible, because downstream, when you think about the antibody, let's say production or an LNP production, both systems, and especially the LNP, is quite complex. So if you combine these two systems, the CMC manufacturing will be more complex, as well as the downstream like stability of these systems will be impacted by like targeting ligand. To your question, which will be harder to target? One example is definitely the kidney. It's not one of the easiest targets. Where I strongly believe in is like that maybe it's not only the LNP which has to do the job, but maybe the LNP has to combine with other technologies which open up the fenestration in the kidney. And we have seen like interesting like physical stimuli where companies or academic teams have used, let's say, high focus ultrasound, for example, to open up different tissues and therefore enable LNPs to accumulate. So I think there's maybe thinking a bit beyond the standard approaches to target the tissues like that. When it comes, maybe one more organ, the lung, we have seen like companies focusing on the lung following intravenous administration, but then you mostly end up in the problem that you don't hit the right cell type, as Dr. Cooke has already outlined nicely. Therefore, it's important to think about the route of administration. Doesn't it make sense when we think about the patient compliance to not do an intravenous administration when you go to the lung, but actually try to really overcome this route of administration like barrier and do a pulmonary administration, what we have seen from many asthma like drugs, that you can actually apply in let's say an LNP mRNA system via the lung. That would be the easiest route. Obviously there are then stability issues with the LNP and you have to overcome other hurdles. But from a patient perspective, this is really the key in my perspective to overcome.
There was an article in Science a couple of years ago that really was galvanizing for the field, coming out of Penn by Drs. Weissman and Epstein and other colleagues. What they did, and you'll recall this, is they generated in vivo CAR T cells. Those cells were directed against activated myofibroblasts in the heart to reduce cardiac fibrosis in that animal model. Really exciting, the idea of in vivo generation of CAR T cells with LNPs that are targeting T cells and carrying the chimeric antigen receptor encoded as mRNA, the really exciting approach. So questions come in from our viewers on that topic. And here's the question related to that background. Does in vivo CAR T cells provide a durable response like the conventional ex vivo engineering? So that's one question. And does this type of drug delivery system going to have likely to have variation in gene expression? So from the audience.
Yeah. First going to the first question in terms of durability, and I think therefore the first step is like to understand for what diseases do we want to use that. And I would say in the field there are two main applications: oncology and using, let's say, CD19 to target like B-cell leukemia, or then autoimmune diseases. And I think to the question of durability, if mRNA approaches are enough, I think we don't have enough knowledge from the human setting in clinical trials to actually answer that at that moment. So the next like outcomes of the clinical trials will show that. Most probably, the field is currently believing that for cancer it might not be durable enough, versus for applications where we think about could be fibrosis, but could be more thinking about like autoimmune diseases, an mRNA approach which is transient could work efficiently, and also by then reinfusing the mRNA LNPs from time to time. So one shot will not obviously be enough, most probably. And then the second question was about variation. Can you outline the question again?
It was about could this drug delivery system, is it likely to have more variation in gene expression?
Like from using different lipid nanoparticles or looking at different cell types?
Yeah, I think the idea is that there's a lot of potential steps between injecting an LNP and getting gene expression in circulating cells.
Yeah, I think what is important is that, first of all, like when we talk about the preclinical setting, we have to understand that there's obviously a difference between mice, NHP, and human. And then thinking about CAR T cell therapy, what is there a certain subtype we have to target in the T cells, or is like generally like all CD4, all CD8 T cells? I think these are key questions which have to be answered. I would align with the statement there will be different expression levels. I'm not sure if that question was really targeted towards like different cell types or also using different types of LNPs. Like obviously you have to like go up with a dose to hit a certain efficacy, but you want to keep the dose low enough to not run into any dose limiting toxicities. So the LNP composition will be a main driver for some of these safety aspects.
You know, one thing that we haven't talked about yet, Dr. Witzigmann, is the immunogenic response, the immunological response to the LNPs. The LNPs have some inflammatory, pro-inflammatory effect. That's useful for a vaccine, not so useful at all if you're trying to use mRNA in a therapeutic way. Tell us about the immunogenicity of your LNP, your long-circulating LNP.
Yeah, and there in our standard screening, when we screen LCLNPs and look at different compositions, the main driver for immunogenicity is mainly the ionizable lipid. And therefore it's really important for us that we have our own lipid library where we can screen different lipids in different types of compositions, and we typically screen like a cytokine panel in each in vivo study that we can understand the immunogenicity of the ionizable lipid and then the LNP. We pick obviously ones which have a low to zero immunogenicity profile, but extending towards that, what we have seen with partners, and as mentioned, we work on sRNA with partners as well as mRNA as well as DNA. The payload is also a key driver for some of the aspects. For example, we had the same LCLNP in a setting where we used it for DNA, and we have seen when the DNA payload was not optimized, there was quite some immune response. When we have used exactly the same composition to show our partner that it's not the delivery system per se, but actually it's driven by the combination of the delivery system with the payload, and use this for mRNA or sRNA, we actually didn't see this immune response. So what I want to say with this, you cannot only think about the LNP, you also have to optimize the payload, which will impact the immunogenicity profile. Mm-hmm. Third aspect beyond this is like, how can we actually combine LNPs maybe with other approaches? The fascinating part of an LNP is it's a modular system. So there have been groups also in Vancouver from Peter Callis and other companies using the LNP as a multi-component system where you can, for example, include, for example, dexamethasone prodrugs in the same LNP. So wherever the LNP is delivered towards, you also get an immune suppression. And these types of like combination therapies, I think, will also overcome some of the critical issues we see.
Mm-hmm. Oh, that's interesting. You showed us one slide that was really striking, and that was the morphology of your LNPs. So you call them the fried egg morphology.
Yeah.
And I was wondering how, what is the mechanism by which that change in morphology causes the LNP to avoid the liver? What's going on there? Do you understand the mechanism?
So first answer is we don't understand it completely. One type of studies we have done is looking at the biomolecular corona, like the protein corona, for example, and we have seen that there are differences in types of proteins as well as the amount of proteins accumulating to the surface. And this is like known from the liposomal field when we look at liposomal doxorubicin, that the accumulation of proteins when we take a pegylated liposomal system or unpegylated, but then using DSPC versus sphingomyelin, you can like change the biomolecular corona of a lipid nanoparticle system or a liposomal system. So one answer to your question is, yes, we see differences. This is part of the answer. If it's the complete answer, I don't think so. So I cannot give you the complete answer to that. What we definitely see is that the morphology, and this was our hypothesis from the start, if we induce this lipid bilayer structure, we have extended circulation half-life independent of any type of helper lipid. So the helper lipids can add an additional effect on that.
Right. Dr. Brannan, Dr. Shah?
I guess one more thing I was thinking of was the advent of some technologies, some delivery technologies that are not necessarily lipid-based, like extracellular vesicles or viral-like particles that are sort of coming onto the stage. And what are your thoughts about the future of how those might compete with or cooperate with the field of lipid nanoparticles?
Yeah, really interesting question, really important question. Like I would say, lipid nanoparticles have proven as like lipid-based systems, like in the liposomal field as well as the on petro, the genome editing field now, not approved yet, but like really successful in the clinic, or COVID-19 vaccines, that they have been able to overcome certain delivery barriers and show the efficacy and safety to get approved. What other systems, like let's say exosomes or viral-like particles or polymer nanoparticles, not have shown yet, and they are for sure behind the LNP field in terms of like the regulatory aspect. And this is one important consideration you need to have, is that the regulatory is complex, and let's say also for the LNP field there needs to be like opening up of the regulatory framework in, when we think about personalized therapies. But polymers, extracellular vesicles have to still overcome that, and also they have to show that how simple they are. And what I mean with this, an LNP, even though it's like a complex system, it's pretty defined. Like DSPC has a specific molecular weight, cholesterol has a specific molecular weight, the iron has a lipid. Versus when you think about polymers, they are not one defined molecule. They typically have like a distribution of molecular weight. And then also when we think about extracellular vesicles, how can you make this, let's say, pure enough that to overcome certain regulatory hurdles? I strongly believe each delivery system will have its niche. I think they are just like the LNPs are ahead of other delivery systems because of the clinical success. Not saying that the others cannot be successful, but I think it will be more challenging. But it's in like from a pharmaceutical technologist point of view, it's super exciting to see all the different technologies. And let's say the more competition there is, the hotter will be the field, and the more we can like open up certain tissues. So I'm excited about all these developments. Obviously biased towards non-viral delivery systems versus viral delivery systems.
I have more questions, but I want to give Dr. Shah a chance if he has a burning question.
Yeah, just a one. I'm very new to this LNP field, and I'm learning along the way, but how complex is it to make LNP with this long circulating LNP with other stuff like nucleic acid, with the drug in closing? And will the size matters when you are delivering it to the organs?
Yeah. So in terms of like combination, I think this is generally when I think about lipid-based systems, what fascinates me that it's, as mentioned, like modular. So we have used the LNP, the LC-LNP in combination with small molecule prodrugs, and looking at the benefit, the added benefit, when especially what Mr. Cooke or Dr. Cooke was asking about immunogenicity. So how can we overcome certain let's say side effects? So the combination is not a problem per se, because what you do during the formulation procedure is you just add other components or take some components out. You do the same formulation method, and what you get out is this modular system which has different components. When it comes to the nucleic acid payloads, there are fine differences when you think about an siRNA, well, fine or major differences when you think about an siRNA versus a self-amplifying RNA. Self-amplifying RNA is significantly larger compared to an mRNA, and even much larger than an siRNA. So when you think about an LNP as roughly like 300 siRNAs or 600 siRNAs per LNP versus maybe only one saRNA, self-amplifying RNA per LNP, there are two main things to consider here. One is you need to adjust the lipid to RNA ratio to have a better encapsulation. But also if you have like, the larger the payload is, the more heterogeneous is the final composite, like the final LNP, because you don't have like 600 siRNAs in each LNP. You have like one, so some might be empty versus in some there might be two or maybe only one. So you have to consider the lipid ratio when you formulate larger payloads compared to smaller ones.
I have two questions for you related directly to some programs that we have going on here at Houston Methodist. The first question is related to an mRNA therapeutic that we've developed for radiation-induced damage to the skin. It's a problem delivering to the skin. I think it's easy, it's right there. But you got the stratum corneum, which is a highly impermeable barrier to anything but the smallest lipophilic molecules. You got any ideas about getting LNP RNA through the skin?
Yeah, I would say that it's a super interesting application you're working on. And let's say, from my perspective, we have worked on using with a company in Europe working on, let's say, the LNP type of delivery systems to target skin via the intravenous route, because our partner was focused on not a topical application. We saw let's say, delivery into the skin, but there's also like then, let's say, delivery to other tissues beyond the skin, like the adipocytes. When I think personally about the skin, like topical application, as you mentioned, is the easiest, or it seems to be the easiest. But then there are like certain disease types where the skin barrier is like limited, and there you can apply the LNPs like topically. When you don't have like a, let's say when you have like a correct barrier, skin barrier, and you want to apply it topically, I think the key will be to combine it with physical methods, as I mentioned, for tissues like the kidney. So thinking about like, could there be microneedle approaches or other approaches where you actually physically overcome the skin barrier and then apply the LNPs, because the LNP will be difficult to translitose through the skin layers and then get into the skin. So yeah, I'm looking forward to all the research you are doing in that, and I think especially for the radiation aspect, it's really key for the cancer patients.
And then another question for you is related to personalized RNA therapeutics, and in this case, personalized mRNA cancer therapeutics, and the idea that we can potentially develop hospital-based systems to deliver with these deployable manufacturing units that are becoming available. Do you have any plans to incorporate your encapsulation techniques into a device, deployable manufacturing unit that can be used in a hospital setting for personalized RNA therapies?
Super interesting field. We like within Innovation are not developing like manufacturing systems per se. Like the manufacturing systems we use are the ones which are also used in larger scale and manufacturing, GMP manufacturing. But we are in contact with various companies who offer like the LNP manufacturing in a box. So there are companies who focus on like how to manufacture in GMP the mRNA in a box, and then also the LNP versus like combining these two boxes. There are several different companies, and like when we look at COVID-19 and the companies involved, like BioNTech, there have been recent super interesting studies where they focused and they were like founded to focus on cancer. Then COVID came up, but they apply now the same principles again to COVID-19 vaccines. So last month, or beginning of last month, I have been at the RNA Summit, where BioNTech presented some of their recent data on individualized cancer vaccines, and super exciting. They use like up to 20 different neoantigens in an LNP, administered intravenously, and depending on the tumor type, and there are currently studies on melanoma or pancreatic cancer, also from Moderna, show really like interesting results and like very positive results. And for some of the applications, the earlier you treat the patient in the let's say cancer development, the better and the more effective is the cancer vaccine. So coming back to your question, we work with partners on cancer vaccines, and we have contact to various different like manufacturing companies. And I strongly believe that this will be one of the hot fields and areas we should work on as a field to really leverage the preventive or even like therapeutic potential of cancer vaccines to have a new like treatment regime together with other therapies like PD-1 blockers in combination.
Wonderful, wonderful. Well, I'll let the last question from my junior colleagues, Dr. Shah, Dr. Brannan, any other questions to ask of our guest? If
I
not,
think you answered all mine. Thank you.
if not, I want to thank you, Dr. Witzigmann, for joining us today and giving us a really inspiring talk on the progress in the lipid nanoparticle field. And congratulations on the work that you're doing at Nanovation. And we look forward to seeing more developments, novel developments from your company going forward. Well, thank you all for joining us today. That was Dr. Dominik Witzigmann of Nanovation, and they've been developing wonderful LNP technologies for nucleic acid delivery. And thanks again, Dr. Witzigmann, for joining us on the show.
Thanks a lot for inviting me. Exciting to talk about RNA therapeutics.